Okay, Assalamualaikum warahmatullahi wabarakatuh. I am Madam Siti Shazwani binti Hussein. Okay, so today we are going to learn about chapter 6, which is mutation. Okay, so mutation is divided into 3 subtopics. For the first one is 6.1 mutation, classification and types. 6.2 gene mutation. Okay, 6.3 chromosomal mutation. Okay, let's see 6.1 first. Okay, for classification and type. Okay, here we have classification and types. Okay, so you have to differentiate between these two. Okay, so learning outcome. Okay, define mutation. Okay, next is to state two types of mutation. Okay, which consists of spontaneous mutation and also induced mutation. Okay, C is to classify two classes of mutation. Okay, so here we have types and two types and also two classes. Okay, so for classes we have gene mutation or called as point mutation, and we have chromosomal mutation. Okay, so D we have defined mutagen, and the last one is to state types of mutagen. Okay, so definition of mutation. Okay, what is mutation? So mutation is a spontaneous change. Okay, so it is a spontaneous change in amount, arrangement or structure of genes or DNA of an organism. Okay. Okay, let's look at this picture here. Okay, here we have a normal DNA sequence. Okay, let's see 1, 2, 3, 4, 5, 6. Okay, the 6th amino acid is actually glutamic acid. Okay, so the codon is CTC. Okay, pair up with GAG. But after mutation occur, okay, let's see amino acid number 6. 1, 2, 3, 4, 5, 6. Okay, here we have valine. Why it changed to valine? Because, okay, base T here is substitute with base A. Okay, so the codon already changed. Okay, so when codon change, okay, it will change the amino acid. So amino acid sequence will change after mutation. Okay, so next let's see. Okay, types of mutation. We have spontaneous mutation and also induced mutation. Okay, so spontaneous mutation is natural changes in DNA structure. So it occurs naturally and it occurs randomly, and it is rare. Compared to induced, it caused by mutagen. Okay, so here it occurs naturally, here it caused by mutagen. Okay, and it occurs in higher rate compared to this. This is rarely occur. Okay, so spontaneous mutation occur naturally without human intervention. Okay, caused by non-disjunction. Later we are going to discuss more about non-disjunction. Okay, due to mutagen in environment. Okay, this mutagen may include cosmic rays, radioactive compound, and also heat. Okay, so it occurs also during DNA replication. Okay, as I said earlier, spontaneous mutation caused by non-disjunction. Okay, so non-disjunction occurs during meiosis. Okay, either meiosis 1 or meiosis 2. Okay, in this diagram, okay, this non-disjunction occurs during meiosis 2. Okay, like what you can see here. Okay, here there are proper disjunction. Okay, but for here, okay, there is non-disjunction. Okay, for proper disjunction, okay, two sister chromatids separate and move towards opposite poles. Okay, but for here, okay, these two sister chromatids are unequally divided, so it called as non-disjunction during meiosis, either meiosis 1 or meiosis 2. But for this case, non-disjunction occurs during meiosis 2. Okay, next. Okay, so errors during DNA replication. Okay, spontaneous mutation also occurs because of errors during DNA replication. Okay, the nucleotide base pair up with non-complementary bases. Okay, but the errors are not corrected by DNA repair system. Okay, let's look at this picture. Okay, this is parental DNA and it undergoes DNA replication. Okay, during DNA replication, okay, this template, okay, G pair up with C. Okay, this is the correct one. But for this template, okay, the G is pairing up with T. G supposedly pair up with C. So after DNA replication occur, it become mutant. But for this template, okay, it is the normal DNA produced, so also called as what? Wild type. Okay, that one is for errors during DNA replication. Okay, for induced mutation, okay, it is also artificial induced and it involves human intervention. Okay, caused by mutagen. Later on, we are going to discuss more about mutagen that cause DNA damage and alter the genes. Okay. Next, physical agent. It involves physical agent, for example, X-rays, UV rays. It also involves chemical agent. For example, we have mustard gas, ethylene amine, and also colchicine. Okay. Induced mutation also helps evolution, and it also helps genetic variation at higher rates. Okay. So now let's move on to definition of mutagen. Okay, so what is mutagen? Mutagen is a chemical or physical agent that can cause mutation, thus increases the rate of mutation. Okay, it can increase the rate of mutation. Okay, so there are two types of mutagen. So we have physical mutagen and also chemical mutagen. Okay, so you have to know at least 3 examples for physical mutagen. So we have UV rays, gamma rays, and X-rays. And at least you have to remember 2 for chemical mutagen. So we have colchicine and ethidium bromide. Okay, so this is the example for physical mutagen, and this diagram is for chemical mutagen. Okay, so next, okay, for the classes of mutation, okay, just now we have discussed about types of mutation, which consists of spontaneous and induced mutation. But for this one, okay, this is class for mutation, okay, or class of mutation. So here also we have two classes of mutation: gene mutation and chromosomal mutation. Okay, for gene mutation, we have 4 types of gene mutation: base substitution, okay, base insertion, base deletion, and base inversion. And for chromosomal mutation, we have chromosome aberration and alternation of chromosome number. Okay, here we have chromosome aberration. Okay, chromosome aberration means that change in structure. So we have translocation, deletion, inversion, and duplication. And also here we have alternation of chromosome number. Okay, let's see chromosome numbers. Okay, here alternation of chromosome number means that the chromosome numbers will be altered. Okay, because of aneuploidy and also euploidy, or called as polyploidy. So polyploidy divided into autopolyploidy and also allopolyploidy. Okay, now we are going to move on to 6.2, which is gene mutation. Okay, so for gene mutation, so the learning outcomes, okay, are to define gene mutation, and also you have to know how to state 4 types of gene mutation. Okay, you have to know 4 types of gene mutation. Okay, so now we look at definition of gene mutation. Okay, a definition of gene mutation is a change in a single nucleotide, okay, nucleotide pair of gene that lead to the change in amino acid sequences. Okay, so gene mutation occurs due to error in base pairing during DNA replication. Okay, look at this diagram. Okay, so here we have, okay, DNA replication. Okay, look at this base pair. Okay, here G is substituted with T. Okay, so when G is substituted with T, So T will pair up with A. So there are error occurs here during DNA replication. Okay, next. Okay, gene mutation. We have 4 types of gene mutation. Okay, the first one is base substitution. Okay, so for base substitution, base substitution occurs when a pair of nucleotide is replaced with another pair of nucleotide. Okay, for example, here we have normal DNA. Okay, so here normal DNA, so it substituted with different pair of nucleotide. Okay, so TA is substituted with G and C. Okay. Alright, so for base insertion, it is an addition of one or a few nucleotide in DNA sequence. Okay, addition of one or a few nucleotide in DNA sequence. Okay, this is the normal DNA. Okay, this is sequence after base insertion. Okay, so here we have A, G, C, T, T, A. Okay, here we have A, G, C, G. So here one addition of base pair with C, which is G. Okay, so base insertion involve addition of one or a few nucleotide in DNA sequence. Okay, for base deletion, deletion means loss or removal. So if there is loss or a removal of one or a few nucleotide in a sequence, it is called as base deletion. Okay, for example, here we have normal DNA. So here A, G, C, T, A. So here one T is deleted. So the sequence become shorter. DNA sequence become shorter. Okay, so for base inversion, for the last one we have base inversion. Okay, so for base inversion, there are two or more nucleotides are inverted and it is rotates 100 degree. Okay, for example here we have A, G, T, C, T, A. Okay, alright, so okay, A, G, T, C, T, A. Here we have A, G, C, T, A, T, A. Okay, so here we have for base inversion, here we have T and C rotate together. Okay, supposedly C, is up here and T is down here. So when there is a rotation, T going up and C going down. So when it is inverted, so T will pair up with A and C will pair up with G. Okay. Okay, so we have finished learning about 6.1 and 6.2. Okay, now try to answer these questions. Okay, so number 1 is to define mutation. Okay, try to define mutation. Okay, try not to refer to your lecture notes. Okay, try to remember the definition of mutation. And second one is to state 4 types of gene mutation. Okay, try to state 4 types of gene mutation.